Okay, I'm back. Sorry about that. We lost some internet up here because we were having some strong winds. So let me go back. We left off talking about this histological specimen of the digestive tract. And as I said last night, this is the lumen. So this is where the food would be going through. And here is the simple columnar epithelium. So letter A is simple columnar epithelium. Most of the digestive tract has simple columnar epithelium from the level of the stomach through the small intestine, through the large intestine until you get to the rectum. Letter B is showing you the lamina propria. Letter C is showing you the muscularis mucosae. And if you need to remind yourself of these layers, it is actually found in your digestive system lecture, specifically slide 14, if you need help with these layers, because I'm just abbreviating them. Letter D is showing you the submucosa. And letters E and F together are the muscularis externi. Specifically, the inner layer is the circular, and the outer layer is the longitudinal. And then letter G is showing you the serosa. All right, moving on. Now this picture is showing you a picture of the stomach, which would have simple columnar epithelium. And we could see the simple columnar cells here with their nuclei in the center. And these are invaginations. I know it looks to you like there are fingers. These are not a finger-like presentation similar to what we're going to see for the small intestine. These are invaginations. So these are gastric pits. And we would find gastric glands in there and pyloric glands. And we would find cells like parietal cells that are releasing intrinsic factor and hydrochloric acid, and chief cells that are releasing pepsinogen. And these cells up here that you're seeing are mucosa cells. And the mucosa cells are very important to release the mucus, which protects the epithelial lining of the stomach from the very harsh environment. The pH in the stomach can get down to as low as 2. That is very acidic. And some people have a problem with ulcers. They get ulcers in their stomach. They get ulcers in their duodenum, which is the first part of the small intestine. And a person can develop ulcers basically either from production of too much acid, or they don't have enough protection from the acid that they are producing. And you'll learn more about that in physiology. I already told you what that was, and I told you the answers to that. So this is now showing you a lobule, a hepatic lobule. That's the functional unit of the liver is the lobule. That's these almost hexagonal looking repeating structures here. And over here, let me highlight that in purple. Over here, I'm pointing to a triad. Okay, a portal triad. And in the triad, we see a bile canaliculus, or a branch of what will become the bile duct. We see a branch of the hepatic artery, and we also see a branch of the hepatic portal vein. In the center here, we see the central vein. So what happens is blood that came from the GI tract percolates through these plates, and all these red dots that you are seeing are hepatocytes. So as the plasma percolates through these large sinuses, remember you learned in Unit 2, you learned that there were 3 different kinds of capillary beds. You learned that the most abundant type was continuous. Then you learned about the about the sinusoidal capillaries, the large venous ones that I was telling you about. Venous, I shouldn't have used that. And then you also learned about the glomerulus, for example, which has the filtration pores. So these are capillary beds that are very porous. They don't have a continuous lamina propria. And the detoxified blood now goes into the central vein, which will ultimately go to the hepatic vein, which then dumps into the inferior vena cava. So I just checked in your digestive system. This would be slide 35 if you want to review the lobule of the liver and the hepatic triad, the hepatic portal triad. And if you want to review the liver sinusoids, all of that is discussed on slide 35. Okay. Yep. Now, this definitely has finger-like projections. And I urge you to go back and compare this slide with the stomach slide that I went over 2 slides ago. These are called villi. Those are the finger-like projections. Again, these clear-looking cells are actually goblet cells that are producing mucin. You can go back to chapter 4 and review what a goblet cell looks like. Again, it looks like a wine glass with a long stem. This dashed arrow is pointing to a Peyer's patch, which is a colony of a single B cell. You can find Peyer's patches not only in the small intestine, which is where this slide is from, but also you can see them in the large intestine. And you learned about Peyer's patch back when you learned about your lymphatic vessels, and you also learned a little bit about immunity. So you can go back to that lecture and review. All right, told you that. Now, here is a picture of the large intestine. These are not finger-like projections, rather very similar to the stomach. These are invaginations. These are intestinal glands, and they're going inside. And look at all the goblet cells that we see here. They look like they're just open spaces because these cells are filled with mucin, and mucin is translucent to light, so the light just passes right through. And here's another Peyer's patch, in fact, down here. I know there isn't an arrow to it, but I can see it right there. The large intestine is a site where you can have vitamin B synthesis. You have some more absorption of water from your diet. And its primary job is to basically be a storage site for fecal material until it's socially acceptable for you to pass it. Okay. And I did say what kind of epithelium you find in the large intestine a few slides back. I said what type of epithelium is found through most of the GI tract, starting with the stomach through the large intestine until you get to the rectum. So I am going to let you go back and figure out what I said. Now, this picture is from the kidney. Specifically, this is a duct. It could be a medullary duct, for example, or it could be the thick ascending limb. It is lined with simple cuboidal cells. We can see how perfectly circular the nuclei are. So those are simple cuboidal epithelium. We're just going to call this a duct. And we would have filtrate going through here. And again, this is from the kidney. And what is the tissue's functional significance? I've also said this before, back when we learned about chapter 4. Whenever we see squamous epithelium, we think of a thin distance when we're talking about simple squamous. And when we're talking about simple cuboidal or simple columnar, now we're talking about getting enough surface area and cell volume to focus on synthesizing and releasing digestive enzymes, for example, or having enough surface area for membrane transport, in this case, from the kidneys to reabsorb electrolytes and water and glucose, etc. Now, these structures here are Bowman's capsule, or glomerular capsule, and this stuff inside is the glomerulus, which is a capillary. So here we see this space, the glomerular space, and filtrate, which comes from plasma, goes from the glomeruli into the glomerular capsule. The glomerulus plus the glomerular capsule, shown here, is called a renal corpuscle. So there you go. I have addressed what these arrows and brackets are pointing to. And the tissue that we find at the end of the solid arrow would be simple squamous epithelium. You can find that information in your urinary system lecture. Specifically, it would be found on, let me find it for you. Specifically, you can find all of this on slide 9. Slide 9 of your urinary system. It tells you what a renal corpuscle is. It consists of the glomerulus and the glomerular capsule. And then the filtration membrane is discussed on slide 10 of your urinary lecture. Now we are in the testis, and in bracket we see seminiferous tubules. And the seminiferous tubules are going to have developing gametes. They're going to have spermatogonia that are going through meiosis 1 and meiosis 2. And as the cells move towards the center of the lumen of the seminiferous tubule, they start to become more and more mature-looking sperm cells. They still have to mature for a few days before they actually are able to swim and fertilize an ovum. But as the cells migrate towards the center, they are becoming more differentiated. Now, these cells in the middle here are called interstitial cells. But I grew up interstitial, I didn't even spell that right. Interstitial cells, but I grew up knowing them as Leydig cells. And there are also support cells inside the seminiferous tubule called Sertoli cells. And your textbook calls those something different if you hold on. See, the old terms we're trying to get away from. So give me a moment and I will tell you what your textbook now calls them. Here we go. Leydig cells and Sertoli cells, you can find this in your reproductive lecture, specifically slide 5. As I said, Leydig cells, they are found in between the seminiferous tubules. They are called interstitial cells. The Sertoli cells are found within the seminiferous tubules, and they are also called sestinacular cells. And the Sertoli cells are going to protect the developing gametes and promote their development. Sestinacular cells respond primarily to FSH, And the interstitial cells respond primarily to luteinizing hormone, and they are the ones that release testosterone in great amounts and minute amounts of estrogen. So again, that was on slide 5 of your reproductive system, if you want to go and check that out. And that will certainly help you answer a lot of these questions, like what hormones do they secrete? What do these hormones do for the body? So you can find that again in your reproductive chapter, but you also can find out what the hormones are going to be doing for the body in the endocrine chapter as well. And then our last histological slide is of the ovary. And there's a lot going on in the ovary. I think female reproductive anatomy is a lot more challenging. And you can find all of the answers to this slide from your reproductive chapter, specifically the cartoon drawing of the ovary. There are 2 of them. There's slide 17 and there's slide 19. So slide 17 has a lot going on, and I'll start to address them. Okay, well, I'm gonna get back to my PowerPoint here. Okay, so these out here in the periphery are primordial follicles. And this bracket is showing you a mature follicle or a Graafian follicle. I think it actually has 3 names. So mature, come on. Mature follicle or a Graafian, I think there's 2 A's, follicle. And I think the textbook has a 3rd name. Let me find it. Vesicular follicle. I was right. Vesicular follicle. Let me write that down. This structure that the dashed arrow is pointing to is a primary oocyte. It is arrested in meiosis I. Specifically in prophase. Now, your textbook says that shortly before ovulation, that primary oocyte will finish meiosis I and then will enter meiosis II and will be arrested in metaphase, metaphase II. However, I want us, for testing purposes, to know that we're going to say that this primary oocyte does not finish meiosis I until it is being ovulated. I just think that's a better landmark for us. And once it's out, when it is ovulated into the fallopian tubes or uterine tubes, then it is in meiosis II and arrested in metaphase II. So it's weird to think, ladies, when we are ovulating a structure that hasn't even finished meiosis. And it's not until it's fertilized that it actually will finish meiosis II. So this structure here that the big solid arrow is pointing to a follicle that is starting to mature. So it's no longer primordial, and there are different stages that the follicles go under for maturation. And again, this is shown on slide 17 of your reproductive chapter. So the primordial follicles are going to start developing into primary follicles, which is what that large arrow was pointing to. And when we start to see a fluid-filled antrum, or an area around the oocyte where we start to see fluid accumulating, then we call it a secondary follicle. And there are cells around the follicle, and cells around the developing oocyte. So the cells around the follicle that I'll point out are called theca folliculi. This is on slide 19 of your reproductive chapter. And then the inner cell area, inner cellular mass, are the granulosa cells. So the theca folliculi cells they're not smooth muscle, but they have smooth muscle-like tendencies. They are the ones that actually apply a pressure to cause the oocyte to erupt. And again, more of this is discussed in your reproductive chapter. That's all sperm. It's in the reproductive chapter, but also the endocrine. Let me pull that up. Endocrine, specifically the endocrine PowerPoint slide 43. So the theca folliculi are going to produce androgens, and then those androgens, like testosterone, if you will, are going to be converted to estrogen by the granulosa cells. So again, that's slide 43 of the endocrine chapter to help you with those cells. So again, out here, let me highlight it in red. So out here in the periphery around the mature follicle would be the theca folliculi cells. And then inside the cells will be more of the granulosa cells. And then there are also granulosa cells all right here around the developing oocyte, sorry. And this is called the corona. They will be released with it. Corona radiata. Okay. Okay. That is the end of our histology.